This is video number 4 in our series about myopia. In this one, we discuss how, for some people, eye growth diverges into myopia. In earlier videos, we have discussed what myopia is, both in terms of the well-known blurry distance vision and the less well-known risks it poses for eye health. Why does myopia happen? To answer that, we have to understand eye growth, which we have divided into two parts. Video 3 explains how the eye controls its own growth by a process called emmetropization, which seems to be quite successful early in eye development. Yet, while most people keep a stable refraction, beginning at age 5 or 6, some eyes start to diverge into myopia, which is the subject of this video. This is our plan for this video. It begins with a review of eye growth and emmetropization. Then we look at eye growth specifically in myopes, as they diverge from the more stable emmetropes and hyperopes. The big question is, why do some eyes diverge from stability? We look at input from genes and the environment. We also take a look at what treatment trial results can tell us about proposed mechanisms. In the end, we attempt to put all this in perspective. One more thing before we start. Keep in mind the big picture. If the goal is to prevent the refractive and health complications of myopia, then we need a way of identifying who will get it and an effective means to treat it. For those who have not seen the previous videos, we start with a brief review of the basics you will need to understand this video. If you have seen the previous videos, you can skip ahead to the next section, Growth into Myopia. If you had ideal vision, light rays from a distant object would come to a focus and make a sharp image on the retina in the back of the eye. The cornea and lens in the front of the eye act together to supply the focusing power. To form a sharp image, the focus distance must be a match to the length of the eye. This ideal is called emmetropia. To the extent eye growth gets off track, the eye can become near or farsighted, usually hyperopia resulting from a too short eye and myopia from a too long eye. At birth, human infants, like many other animals, start out on average as hyperopic and progress toward emmetropia. The direction of growth is guided by a process called emmetropization. As the eye grows, the focusing power of the front of the eye gradually changes along a pre-programmed course. The mechanism for keeping the image in focus on the retina depends on fine-tuning the length of the eye. Surprisingly, it is not the axial focus where you perceive sharpness that controls emmetropization. Rather, it is the image from the mid-periphery, where the retina, remarkably, is able to detect whether the image is out of focus and which direction it is out of focus. In the growing eye with hyperopic defocus, the eye generates a go signal for increase in length. The same go signal can be triggered by using a minus lens to induce hyperopic defocus or by form deprivation. For myopic defocus, the eye generates a stop signal, and growth slows, allowing the image to move back to the retina. The same stop signal can be triggered by using a plus lens to induce myopic defocus. A red light or bright light may also generate a stop signal. By whatever mechanism the retina detects direction of defocus, there is a cascade of signaling within the eye that directs the sclera to remodel, thus controlling the length of the eye and refractive status. That is emmetropization in one simple diagram. Video 3 covers this in detail. The success of this mechanism can be seen in the narrow distribution of refractive errors in adults. Whereas, if something blocks a clear image and emmetropization can't work, the result, the orange line, shows that without a clear image, feedback control is lost, and the eye takes on a wide range of myopia, up to -14 diopters in this survey. Note that the result is myopia, not hyperopia. If you look at emmetropization from the point of view of whole population figures and animal models, it appears near perfectly successful. But, as I mentioned at the outset, while myopes start near emmetropia, they gradually diverge from that initial successful result. That divergence has become a big deal, as there is a worldwide trend toward increasing myopia, becoming epidemic in some regions. For example, U.S. figures for myopia show an increase from the early 1970s, the blue line, to the turn of the century, the orange line, which is approaching 50% of the population. In East Asian countries, myopia prevalence is increasing even more rapidly. The top line is 100%, and they are using the term epidemic of myopia. Thus, we arrive at the subject of this video. Where does myopia come from? We are ready to proceed into how the eye grows into myopia. In the big picture, over a lifetime, we see refraction divides into three groups with different behaviors. Emmetropes hold a very steady course over a lifetime. Hyperopes have more variability, but end up close to where they started. While myopes start near emmetropia, they diverge from the other groups somewhere between age 5 to 10, and myopia gradually increases until it appears to level out between 20 to 30. This is the age range we are interested in when myopia is developing, before other factors like the lens's loss of flexibility and cataract changes kick in. What is it that happens to the myopia group in childhood? Has emmetropization stopped working, or become reduced in function, and/or has some other stimulus taken over control? To answer that question, let's start with a look at refraction in animals, particularly monkeys, for what they can tell us about normal development. In the previous video, we saw examples of how refraction changes in other animals early in life. In chicks, correction occurs within the first 3 months of life. In tree shrews, it occurs in the month after eye opening. And in monkeys, correction occurs mostly in the first 3 months, then holds stable. It's easier to see with a single typical monkey. This early correction is attributed to emmetropization. Does it continue working after the first few months of life? Consider this result from a study that used an experimental lens to induce myopia by moving the image focus. When the experimental lens was removed, the myopic eye recovered to near emmetropia, meaning emmetropization was still an active process. However, this was at less than 300 days of age. We need to spend some more time with monkeys. Is this a useful direction? The answer is yes. Significant effort has gone into documenting that the components of monkey eye refractive development are very similar to those in humans. The high degree of similarity will lead us to an interesting comparison. This group studied normal refractive development in rhesus monkeys. Just as we saw in humans, newborn monkeys are hyperopic. This is how monkey refraction tracks over 4 years. For the group as a whole, the average refraction started out hyperopic and steadily headed toward emmetropia, reaching 90% of its final refraction at about 1,000 days. As part of the study, they followed 10 individual monkeys for the full time, their refractive course shown by the dashed lines. At the end, the spread of refractions was not large, with 1 mild to moderate hyperope and no significant myopes. But this ends at monkey age 4 years. We can go further. This group measured refractive error in macaque monkeys over their entire lifetime, the average lifetime being about 27 years, though some can make 35. Relating that to humans, researchers scale monkey lifetime to human overall at a ratio of 1 to 3, meaning a monkey at age 20 equals a human at age 60. Now, looking at the big picture in monkeys, The range of refractive errors is small. 95% of refractions between about plano and about +5. Most of the myopia did not develop until after monkey age 15, entering their middle age. For monkeys, other age-related effects like cataract become more frequent after age 15. The period we are interested in, human youth up to age 30, equals monkey youth up to age 10. Expanding that section of the graph shows a few hyperopes and no myopes. Looking at the equivalent age groups side by side, for the monkeys, we do not know their history of light exposure. Presumably they have little schoolwork and are unlikely to have glasses. Thus, to the extent we can connect monkey eye with human eye development, this could be taken as the direction of human eye development without the influence of myopogenic environmental factors. What a compelling thought. We move back to humans now. For humans, we start by looking at refractive development in children aged 3 to 10 years. In this survey, at age 3, 80% of the children were emmetropic. Less than 2% were myopic. That percentage holds through age 6. Then something happens. From age 6 through 10, emmetropia drops to under 50%, while myopia correspondingly increases. We switch to a similar survey by Jones, moving to a slightly later time frame, children ages 6 to 14. This is the technical description of each group. Those children who start as emmetropes, just on the hyperopic side, remain stable over time. Though hyperopes can be divided into two categories, in general they remain in a relatively narrow range. The most interesting group are the myopes. Emmetropes and myopes both started out close together, near zero. Emmetropes changed very little, while myopes progressively increased in degree. The authors asked, what was the source of the refractive change within each group? To do this, they measured the changes in the components of focusing power and axial length. Regarding focusing power, myopes started with somewhat steeper corneas than hyperopes and stayed steady, while emmetropes decreased. Lens power started at about the same and decreased a little more in myopes. Thus, in myopes, the steeper cornea but less powerful lens should tend to offset each other. Regarding axial length, myopes and emmetropes started out at about the same point, with longer eyes than persistent hyperopes. While the other groups lengthened the same amount, myopes increased significantly more. Here we go a step further by combining the components for each refractive group to give us a picture of how the various components work together. In this illustration, myopic refraction is downward. For emmetropes, the cornea and lens power gradually decline while axial length increases. These are synchronized so that refraction stays stable near zero. Overlaying the persistent hyperopes shows the components follow a similar path and the refraction is stable. It just started at different places. What about the myopes? Relative to emmetropes, corneal power remains higher while lens power decreases, which tends to offset each other. The big change is axial length, increasing off the top of the chart, while myopic refraction descends off the bottom of the chart. There is more discussion of details in the paper, but the conclusion is that axial length is the controlling factor in myopia development. And this section gives us a nice feel for the relative growth of the different parts of the eye. We are going to come back to this subject in a few minutes. From the last graph, you might get the impression that myopes diverge from emmetropia as a group at the same age. But that is not the case. This family of curves shows the onset and evolution of myopia in children. They pass the threshold of myopia at a range of ages. Also, the age of onset has consequences. First, the earlier myopia starts, the higher degree of myopia at the end. Second, the rate of change, the slope of the curve, is different with different age of onset. As a practical matter, in making a treatment decision, if your aim is to prevent myopia over 3 diopters, according to this chart, you would treat children who developed myopia at age 8 or younger. Here is another version of the same graph strategy, but the age range is 8 to 17 years. In this study, if myopia started at age 8, by age 17 the average refraction was over -6 diopters. Looking at their results another way, the chance of developing high myopia, defined as greater than -6 diopters, for those whose myopia started at age 7 or 8, the risk was greater than 50%. So, age of onset of myopia is an important, perhaps the key deciding factor that we will talk about in the treatment video. Now we ask, when does the trend toward myopia actually start? These growth curves have shown us an important fact, that both age of myopia onset and rate of progression vary. But there is another thing to think about. The eyes were not just stationary at emetropia and then abruptly decided to detour into myopia. What happens before crossing the myopia threshold? Consider this set of curves plotting age versus refraction status for 7 to 13-year-olds. Emetropes, the dark line, remain near zero correction. The red line represents children who were myopic before the observation period. The colored lines in the middle represent children who became myopic during the study, grouped by the age they passed the myopia threshold. That age is noted by the solid circles. So there isn't an abrupt detour into myopia, rather a gradual ascent. The intermediate color lines all look like they originate at a starting point shared with emetropes. Regarding that separation from emetropia, this study from the CLEERE group is going to take us a step deeper. It offers us several interesting results that will take time to present. If this gets too technical, you can skip ahead to the halfway recap. It will be summarized there. For the myopes, they only included children who became myopic during the study. They start in familiar territory comparing the courses of myopia versus emetropia. Here, the x-axis is not age. Rather, this study is based on the time of myopia onset, which is taken as the first visit greater than -0.75 diopters. Note, at 5 and 4 years before myopia, both groups start very close together, about half a diopter hyperopic. By year +5, hyperopes change only about 1/4 of a diopter, while myopes reached an average of -3.8 diopters. The next step is to look at the rate of refraction change, measuring the difference between visits. Here they graphed refraction change comparing myope versus emetrope. Since emetropes changed very little, this essentially shows the yearly change in myopia. Early on, rate of change in refraction was minimal. Then, at 2 to 3 years out, it increased to a moderate level. Clearly, the rate of change peaked in the year before passing the myopia threshold, then settled into a moderate rate for the next 5 years. One additional note. I've labeled the rate of change of half a diopter per year as a reference point. That is usually taken as the average yearly change for myopes. The average yearly change here is approximately 0.4 diopters per year. The 0.5 figure per year will come up again in the treatment video. Axial length is greater in the myopes, as expected. Regarding the rate of change, it behaves similarly to refractive error, starting to ramp up 2 to 3 years out, then peaking in the year before passing into myopia. We mentioned earlier that peripheral refraction is thought to be the guide for emetropization. This is the peripheral refraction measured at 30 degrees nasal to the fovea. It is given as the refraction relative to the fovea. Early on, both groups are myopic relative to the fovea, and the emmetropes stay there the whole time. The myopes start slightly less myopic, becoming peripherally hyperopic just 1 year before conversion. As before, they plotted rate of change of peripheral refraction. The rate of change also peaks in the year before crossing the myopia threshold. Combining the graphs of refraction and axial length confirms the trends we saw before, but lacking cornea and lens data. Combining the rate of change graphs, all of them show changes began several years earlier, peaking in the year before reaching myopia, a very neatly synchronized set of curves. Some studies take the criteria of myopia as -50. That shifts the myopia crossover point a year earlier. Then all the peaks fall just after myopia onset, which doesn't really change the conclusion that the rate of change ramps up several years before crossing the myopia threshold. So, can we tell when the eye begins its detour toward myopia? Here, comparing refractive error and rate of change. When does the eye start its takeoff toward myopia? It looks like at -4 years, myopes are beginning to separate from emmetropes. What causes that takeoff? If peripheral refraction is the driver of myopia, let's look at it relative to central refraction. Is the peripheral refraction in hyperopic defocus as theorized? Interestingly, peripheral refraction starts out myopic relative to the fovea, though it is less myopic than hyperopes. It does not cross into hyperopia until the refraction is well on the path to myopia. Also, peripheral refraction appears to have distinct phases. Minimal change early on, followed by a steep rise to myopia crossover, then leveling off for several years, ending up with a late drop-off. All during a continuous increase in myopia. If you take the myopia curve as relatively smooth, then how does that result from the apparent phases of the peripheral refraction? If instead you use the change in refraction and take it as two main phases, it is still a challenge to devise one consistent mechanism associating the two, let alone understanding the behavior of peripheral refraction. There is speculation about structural restraints inside the eye that could shape this curve. There is still work to be done here. Staying with the same article for one more line of inquiry. This trial specifically aimed to include multiple ethnicities in the myopia question. They presented the next data as difference between myopes and emmetropes. Regarding refraction, interestingly, before crossing the threshold of myopia, there was no difference between ethnic groups. But after the threshold, there emerged significant differences, greatest in Asians. Is it a result of inheritance or environment? Regarding peripheral refraction, there was a greater mix of differences. Asians consistently were more hyperopic, while blacks hovered around zero difference. What we know from this study is that the trajectory for myopia begins well before refraction officially declares myopia. We know from other sources that peripheral refraction is one key driver of emmetropization. It is not yet clear how that connects to myopia development. Earlier on, we talked about the big picture, in which myopia mostly stabilizes in the 20s, though not for all. This study from Northern Ireland carried on human measurements from childhood into the early 20s. From ages 7 to 16, the results were essentially the same as what we've seen so far. Emmetropes and hyperopes remained stable, while myopes showed a progressive increase. From ages 13 to 22, that trend continued, but the progression of myopia slowed, and myopes have been divided into two groups, low and high progressing. Overall, this is like the other families of curves, showing the rate of myopia increase slows over time. This later age range is a good place to point out that some myopes may continue to progress through the 20s and beyond. As we finish the first half about eyeball growth, it is a good time to recap before going on to the search for causes. Early on, emmetropization is successful, resulting in a high percentage of emmetropia. Around age 5 to 6, one group starts to diverge into myopia, eventually comprising almost half the population. Refraction groups all started in a similar range, mildly hyperopic. Hyperopes and emmetropes had similar, mostly stable growth curves, but different starting points, in contrast to myopes, who diverged from emmetropia. Myopes could then be further divided according to age of onset, earlier onset associated with a faster rate of change and higher eventual level of myopia. Digging a little deeper, we saw that the eventually myopic eye started in the same place as emmetropes and began to progress toward myopia several years before crossing the threshold. So whatever mechanism is active started early, years before diagnosis of myopia. Just so that is clear, the myopia groups start to diverge from emmetropia some years before crossing the myopia threshold. A point to consider. If school begins at age 6, the divergence of the early onset myopes begins years ahead of reading and near work. With that, we complete the first half of our inquiry into growth of the eye into myopia. In the second half, we explore why the eye might follow that path, involving genetic and environmental influences. Our exploration of possible causes of myopia starts with genetic background. In video 1, we took a longer look at inheritance. We will briefly summarize that here, starting with just genetics studies. Twin studies showed a high degree of heritability. For a long time, that was the principal evidence for considering myopia to be mostly inherited. In recent decades, there has been a search for specific myopia-related genetic defects. Up to 2010, linkage studies had identified 20 such genes. Keep in mind, though, these are uncommon individual genetic defects that should be considered separate from the general population of myopia. The identification of myopia-associated genes increased significantly with the more recent genome-wide studies. Combining genome-wide studies into a meta-analysis increases the available data set. Just such a study was done for myopia, the CLEERE study. Screening 2.5 million markers, they determined which genes had the highest association with myopia and ranked the strength of association. These were the 19 genes with the highest plausibility scores. What do these genes do? The authors created a nice graphic showing where these specific genes are functioning and how widely distributed the various genes are. However, for complex traits like height or myopia, identified markers generally only account for a small fraction of the phenotypic variation expected from twin studies. The 2018 CLEERE paper, even combined with the 23andMe datasets, only accounted for about 8% of the variation in refraction. That compares to twin studies, which found around 80% heritability. That large gap in strength of association has become known as the missing heritability problem, raising a debate about the relative roles of genes, epigenetics, and environment. It now appears that genes have considerably less influence than was previously thought. In a recent review, one authority concludes the genetic contribution appears to be far more complex than researchers expected. Which brings us to the other side of the coin, environment. We are going to discuss this in two parts. One will be general population studies. The other will be a look at what factors in the environment could be causing myopia. This was covered in more detail in video 1. In 2009, he and co-workers approached the environment question by surveying children from different cities within China. So it is presumed they have similar genetic background. Any difference in results would be from urban versus rural environment. The urban cities showed similar high rates of myopia compared to the lower rates of myopia in the rural environment. The authors concluded the urban-rural difference is likely the result of differences in environmental factors, possibly including educational, socioeconomic, and/or nutritional effects. A similar point is made by Morgan, looking at different ethnic groups in Singapore. They assembled studies to compare myopia in different ethnic groups within Singapore versus the same ethnic groups outside of Singapore. The large difference in prevalence adds to the case for environmental influence. The next question, then, is what is it in the environment that causes myopia? Number of myopic parents provides the background, but is not the full answer. In the environment, multiple studies agree about the effect of sunlight exposure. Findings about near work, curiously, have been mixed. The SAW study we presented earlier found an association with parental history of myopia, reading more than 2 books per week, and other factors listed here. Looking at similar factors, Jones found both parental myopia and hours spent in sports were significantly associated with myopia development. Reading hours showed possible association, but dropped out statistically when corrected for the first 2. Also, Jones found a lack of association with TV and computer time. Jones presented this result showing the effect of number of myopic parents and amount of outdoor time. A child with 2 myopic parents who spends little time outdoors has a 60% chance of becoming myopic. However, if that child with 2 myopic parents spends 14 hours a week outside, the risk drops significantly to the same level as a child who had only 1 myopic parent, and below the risk for the other ones who don't exercise. Also note that having 0 parents with myopia does not mean 0 risk for the child. Indeed, in the low exercise group, the risk is about 30%, the same as having 1 myopic parent. In the Sydney myopia study, they looked for a connection between myopia and multiple risk factors. First, having more parents with myopia is associated with more myopia in children, which fits our expectations. Again, having no parents with myopia does not mean 0 risk. Next is outdoor activity. On this graph, the y-axis is refraction in plus numbers, so hyperopia increases toward the top, myopia is off the bottom. For both age groups, there is a significant trend. Higher amount of outdoor time with a higher level of hyperopia, which is equal to saying higher outdoor activity results in less myopia. Regarding near work, the x-axis is hours of near work broken into 3 levels. In this study, hours of near work did not show a significant correlation with refraction. In addition to the main conclusions, the authors made a couple of additional comments. It was likely that outdoor time was the controlling factor, not sports participation. Indoor sports made no difference in outcome. Second, in addition to the lack of correlation with near activity, they also looked at mid-range activity, TV, video games, and computer time, finding no relation with the final refraction. In 2019, prominent researchers Zadnik and Mutti published an editorial on their views of myopia causes, near work versus sunlight. Consider this as one side of an expert point-counterpoint argument. Zadnik and Mutti identify outdoor time as important, specifically referencing the 2007 Jones graph. They do not find a convincing case for near work as a major cause of myopia. But puzzling that light exposure does not slow myopia once it is present, referencing the following 2017 meta-analysis, which found that outdoor time is effective at reducing onset of myopia, but does not appear to slow development in children who are already myopic. Note This is before the 2018 Wu result. For the counterpoint view on reading and near work, we present this review by Morgan et al. In a survey of the world's literature, they found that educational systems that require a large amount of hours spent in studying and near work and allow for fewer hours of outside time show a strong relation to increased myopia development. A similar study was done in Israel among 22,000 male recruits for military service. The school system in Israel is divided into levels which require different amounts of intensity and near work, with ultra-Orthodox schools requiring the heaviest amount of reading from an early age. Relating prevalence and degree of myopia to school type, clearly the ultra-Orthodox schools had a greater degree of myopia overall, borne out by prevalence and odds ratios. Whether this is from more hours of reading or less time of outdoors, or both, is not addressed in this study. There is one more factor in contention for an environmental cause of myopia development. In the beginning, we discussed that eyeball growth is controlled by a process called emmetropization, which is controlled by image focus, not in the fovea, but in the mid-periphery. Specifically, peripheral hyperopic defocus is a leading candidate for triggering increased eye growth/myopia. That is an important concept because standard glasses focus the image on the fovea, but may leave the peripheral retina hyperopic. This has become a major avenue of investigation for controlling myopia, motivating trials of multiple versions of glasses and contact lenses that aim to control the peripheral refraction. In an attempt to sort out causative factors, a large multi-center study was instituted, the CLEERE study. It used 5 trial sites with more than 4,500 children enrolled. In brief, they looked at 13 different likely factors to try and identify if any could be used to predict development of myopia. Of the 10 that had a detectable relation to myopia development, 8 survived multivariate models. The author's conclusion was, the best predictor of myopia development was baseline refractive error. That single parameter had nearly the equivalent predictive strength of the other 8 variables put together. Following this logic, they suggested a decision to treat could be based on early refractive error. As we discussed in video 3, there is a lot of experimental work behind theories of emmetropization. Anatomically lesioning various parts of the visual system localize the mechanism to within the eye. The ability to induce hyperopia or myopia in animals has taught us a lot about how emmetropization works. Also, light brightness and color have a significant effect on myopia. Again, that is covered in detail in video 3. The Knights have a couple more interesting observations. It turns out the L to M cone ratio, red to green, varies in different populations. As a significant example, the ratio in Caucasians is about 1.7 to 1, whereas in Asians it is about 1 to 1. There is data correlating the L to M ratio with average axial lengths. When red cones dominate, there is more likely to be emmetropia, while if the ratio is even, there is more likely to be myopia. They cite this as one possible explanation of why myopia is more common in Asia than Europe or the US. Another observation involves the haplotype of the L and M opsins. These genes are on the X chromosome with only one active copy of each gene. Looking at exons 3 and 4, sequence variations in these exons determine how well the cone pigments function. The graph on the right shows that variations in these sequences have been found to be associated with specific levels of myopia. In 2015, based on these observations, the Knights filed a patent using both these approaches for diagnostic and treatment proposals. First proposal is to measure the LM ratio and/or determine the haplotype to predict who would develop myopia. Second, use those findings to prescribe a lens with a wavelength-dependent optical filter, or prescribe blur-inducing lenses, either as contacts or glasses. The blur-inducing lenses are currently undergoing a 3-year trial, discussed in the treatment section. In this section, we cover the highlights of treatment trials, the subject of video 5. It is included here because the success or failure of an intervention helps confirm the mechanism it is targeting. If you are going to watch video 5 on treatment, you can watch this as a preview or overview, or you can skip ahead to the perspective section. These are the leading contenders for the causes of myopia, and these would be the routes by which they are addressed. Treatment options include various glasses and contact lens choices, orthokeratology, therapeutic drops, and increased light exposure. The following is a set of highlights covered in much more detail in the video on treatment options. We start with the simplest intervention, glasses options. On the left is a standard single vision lens we are accustomed to correct distance vision. In the rest of the lenses, the orange color represents plus power, an area used for reading. The simplest refractive strategy addresses the theory of hyperopic defocus directly by simply undercorrecting the myopia, leaving the eye intentionally myopic. In theory, myopic defocus should result in slower growth. In one such study, the result of undercorrection was a surprise. The undercorrected eyes, the gold line, developed more myopia than the fully corrected eyes. More myopia. Other undercorrection studies have had mixed results. The second treatment strategy starts with the simple idea that if reading and near work are the cause of myopia, it is logical to look at the effort of accommodation as the cause. Therefore, if you use reading glasses or bifocals to reduce or eliminate the effort of accommodation, that should reduce the development of myopia. The well-known COMET study used progressive bifocals. This resulted in a slight reduction in myopia in the first year of 1/4 diopter and no change after that. The authors commented that, while this can be measured as a true effect, clinically it is not significant and should not be used. Other bifocal studies have shown mixed results. Option 3. Standard glasses focus the image on the fovea, but may leave the peripheral image hyperopic. This has motivated trials of multiple versions of glasses and contact lenses that aim to control the peripheral refraction. About a decade ago, there was a notable trial using this strategy with 3 design variations, differences in central aperture size, and plus power. The study was designed to go for 2 years, but it was stopped after 12 months because of the overall lack of positive effect. Compared to the controls, 2 lens designs showed no improvement and 1 only a small improvement. However, a later trial used glasses that corrected peripheral hyperopia with a different lens design. This trial had more success. Option 4. Increased outdoor time and exposure to sunlight. The He study added 40 minutes of outdoor time on each school day and encouraged more outdoor time in general. In terms of refraction change, there was a small but statistically significant slowing of myopia progress, while for axial length, the change fell just below statistical significance. That gave us a look at the methods we might have expected to work based on our understanding of emmetropization. But that is not all. There are 2 more methods, ortho-K contact lenses and atropine drops. Orthokeratology uses a lens made of a rigid plastic that temporarily molds the cornea with the purpose of changing the glasses prescription. Its effect on myopia development is thought to be through an effect on the peripheral refraction. It has had moderate success. The main pharmacologic treatment for myopia is atropine. It has shown significant success in slowing myopia development. An interesting finding is that the low concentration seems to work the best. Video 5 covers a larger range of studies and has a detailed comparison of their results. For the purpose of this video, in 2016, Wang led a review of the effectiveness of various treatment methods, finding atropine to be the most effective. Again, this is reviewed in detail in Video 5. We have come a long way in exploring causes of myopia. In this last section, we're going to try and put all of this in perspective. For the causes of myopia, we are looking at inheritance as background, modified by other factors in the environment. For a long time, the results of twin studies led to the thought that genes played the dominant role in myopia development. But newer genetic studies find this is not the case. Indeed, the rapid rise in myopia cannot happen from genes alone. The increase must come from changes in environmental factors. Here lie the fundamental causes that can be addressed in treatment. Referring back to the monkey versus human refraction comparison, monkey eye development is very similar to humans, yet in the same period of life, monkeys do not seem to develop myopia. Their light exposure is unknown, but presumably they have little schoolwork and are unlikely to be wearing glasses. For a broad view, we return to the 2018 Morgan Review of Myopia Causes. Note the assembly of all-star authors. Here is how these authors analyze the world increase in myopia. There is an epidemic of myopia in Asia with attendant high myopia. High myopia having potential sight-threatening consequences. Because this increase has occurred over a relatively short time period, it must be acquired, not genetic. They contend the contributing factors appear to be heavy emphasis on near focus and limited time spent outdoors. These risk factors should be modifiable. There are additional measures, for example atropine, that can be used to control myopia. A year later, Morgan and Rose provided a critical review of the genes versus environment debate, including historical background, study methods and conclusions, and current study results. They discussed the debate about nature versus nurture. First, environmental factors can have a major impact, while genetic contributions are more limited than was assumed from twin studies. Second, the idea that myopia primarily occurs due to a genetic failure of control over refractive development needs replacement. It is therefore suggested that school myopia should be regarded as the result of a control mechanism operating in conditions that are so unnatural that myopia occurs as a disease of advanced civilization. With that, we come to the end of how the eye grows into myopia. In the beginning, we saw that emmetropization works well until age 5 or 6. Then one group begins to diverge into myopia. That steadily increases in degree, while the others remain relatively stable. Looking more closely, the divergence from emmetropia actually begins years before crossing the myopia threshold. And all myopes do not emerge at once. Rather, onset happens at different ages. The earlier the start, the higher level they end up at. Inheritance provides the background. The course can be modified by sunlight exposure. Probably near work, and possibly glasses correction with peripheral hyperopic defocus. Each of those is connected to a specific mechanism of myopia development. From that understanding of causative factors comes a range of treatment options. All of that background gives us a much better idea of how to approach myopia, which directly leads to Video 5, Treatment of myopia. That brings us to the end of part 4 in our series exploring myopia. In video number 1, we discussed what myopia was, how it is increasing around the world, and what factors it might be associated with. Video number 2 is a discussion of how myopia leads to vision-threatening complications. In video 3, we tried to understand how the eye is able to control its own growth. Yet, despite the control mechanism, some people diverge into myopia. Last is video number 5, exploring the variety of treatment options available, how successful they are in trials, and how they are used in practice.